Welcome to the ASHI Sensitization in Transplantation Assessment of Risk Working Group 2025 Reports and Planning. Today's session is the first of two webinars for the working group and will feature presentations and discussion on non-HLA antibodies, harmonizing efficacy of antibody reduction therapy using titers, and epitopes, eplets, and transplantation. Attendees are encouraged to share their questions and feedback as the group plans for a pre-meeting event at AST's Cutting Edge of Transplantation meeting in 2025. STAR working group projects have been proudly supported by AST, ASHI, Mark Terasaki, One Lambda, a brand of Thermo Fisher, and Imokar Werfen. In a moment, I will turn the session over to our moderators, Dr. Carrie Schinstock, Professor of Medicine at the Mayo Clinic, and Dr. Anat Tambur, Research Professor of Surgery, Organ Transplantation at Northwestern University. However, before we begin the main presentation, we do have a few housekeeping notes to help you engage with today's session. There is currently a viewership polling question displayed for the audience. Please take a moment to answer this question while we finish the remaining announcements. This webinar is being recorded, and the archive of both webinars will be available approximately 1 week after the live sessions. Attendees are encouraged to participate today, but please note that your lines have all been muted, so only the panelists and presenters can be heard clearly for the live and archive recording. If you do have a question for our panelists during the webinar, we encourage you to participate by using the Q&A button in your Zoom webinar panel. Questions submitted via the chat window may be missed during the presentation. Finally, this is a quick reminder that Day 2 presentations will take place on Thursday, October 10th at the same time, from 1 p.m. Eastern Time to 2:30 p.m. Eastern Time. All registrants for Day 1 will automatically be emailed a brand new login link for tomorrow's Day 2 for the Thursday Day 2 webinar, excuse me. It is now my pleasure to turn the session over to our moderators, Dr. Schinstock and Tambur, to begin our presentation.
Hello, everyone. Thank you for joining us today. It's a great pleasure to come back to another STAR iteration, STAR 2025. My name is Anat Tambur from Northwestern University. I am the founder of the STAR initiative, and I am joined here by my co-moderator and co-chair of the STAR 2025, Carrie Schinstock. I just wanted to, from the bottom of the heart, really acknowledge the support that we're getting from the American Society of Transplantation, the American Society of Histocompatibility and Immunogenetics, and special thanks to Mark Terasaki, who has been supporting this process from the get-go and actually giving us the seed funds to start all of this. This webinar, this STAR iteration, in difference to previous years, will feature two wet lab components as well. And I'm grateful for One Lambda, a brand of Thermo Fisher, and to Werfen Imokar for providing us the reagents to run those wet lab components. With that, I definitely want to thank all the STAR working group members, but special thank you for those labs who had volunteered their technologies to help us perform those wet lab components. I think with this, I will give the microphone to Carrie. Take it away.
Thank you. Really excited about this today. Again, I'm Carrie Schinstock from the Mayo Clinic in Rochester, Minnesota. Just a reminder, after each of the presentations, we'll have a short question and answer. We will have access to all the questions and answers later on. So if we're not able to get to them during the discussion, we're certainly going to keep these comments in mind. But I know we all came here today to hear these presentations, and I'm really delighted to introduce Dr. Michelle Hickey from UCLA Immunogenetics Center, and she's going to be presenting on behalf of her working group, and her talk is entitled Non-HLA Updates and Next Steps.
Thank you, Carrie and Anat. Good morning, everybody, or good afternoon to you from wherever you're joining from. It's nice to see you all here today, and I'm excited to share our update from the STAR Non-HLA Antibody Subcommittee. As a matter of business, I have a conflict of interest to disclose. And I would like to begin by acknowledging all of the members of the Non-HLA subcommittee for the STAR 2025 term. I'm joined during this term by my co-chair, Chris Wiebe, as well as by Annette Jackson, Alex Sinev, Edoya Gemfarar, and we are supported by mentors Elaine Reed and Anita Chong, and statistical support by David Jertson. I just want to give a shout out to all of these members for the hard work and expertise during the past year that brings us to today. And I wanted to begin by reminding everyone that our work builds on the work from the prior term of STAR, which was STAR 2022. In STAR 2022, our goals were to perform a literature review and assess the quality of the literature with respect to analytic and clinical validity of using the non-HLA antibody assays. Our deliverables were published in the STAR 22 publication in AJT, and our deliverables were a literature review, a presentation of the technical characteristics of non-HLA antibody detection assays, organ-specific recommendations for testing, and a list of gaps in the field and recommendations for how to address. This is the organ-specific recommendations for non-HLA antibody testing that was published in the 2023 publication. The table is very detailed, but in a nutshell, we provide recommendations for transplant patients receiving heart, lung, kidney, or liver transplant. There were not sufficient evidence or studies in the literature to grade intestinal, pancreas, or multivisceral transplants. Our recommendations address the testing for non-HLA antibodies in the pre- and post-transplant settings and are presented according to the proposed scheme from the STAR 2017 publication. We found that the quality of evidence in support of testing for AT1R or ETAR or MIC-A, or in using the non-HLA antibody XM1 cell-based cross-match was generally low, indicated by the letter C or D, and ranged between either suggest or do not recommend to test, as indicated with a 2 or a 3. In some scenarios, we based the need for testing on expert opinion. For example, we suggest that testing is reasonable in certain settings, such as post-transplant patients with immediate early graft dysfunction and no HLA donor-specific antibody, or in the setting of heart-lung allograft rejection in the absence of HLA donor-specific antibody. The primary reason for the quality of evidence being considered low is due to significant variability in the quality of the literature, which generally lacks longitudinal antibody testing and serum samples matched to biopsies, or large enough cohorts to account for multivariable statistical modeling. But this said, there are several lines of evidence that suggest that the study of non-HLA antibodies warrants further study to determine if they may be considered as biomarkers of an immune response or as having potentially causative effects on graft dysfunction. So that said, the STAR 22 recommendations were to have continued collaboration with industry to develop standardized reagents and improved quality control, thresholds, metrics, reproducibility, and proficiency testing, and as well standardized reporting between laboratories and studies. We also recommended to determine the clinical utility of non-HLA antibody testing and continued discovery and mechanistic studies. But in the STAR 2025 term, we are focusing our efforts in the area of continued collaboration to further understand the analytic validity of the Luminex reagents that are used to identify non-HLA antibodies. And when you think about the continuum from discovery to the path to the bedside, this is actually one of the earlier steps in the phases. So in the discovery phase, we are working on hypothesis generation and test development. And we could go all the way to clinical utility on the right, which is asking questions. Does the test lead to improved outcomes, and how should the test be used and practiced? But we are focusing during STAR 2025 in the analytic validity box, asking questions about test performance and how well does the test measure what it claims to measure. And this is what brings us to the work of STAR 2025. These are our deliverables. The first is to add to the literature review. And the second is to perform a multi-center comparison of the Luminex reagents from the two vendors. So with respect to our literature review, we have performed an update to identify relevant papers using the same search terms that were used in the prior term of STAR, which are non-HLA antibody and transplantation. And we also performed ad hoc searches, and we collated papers from the years 2021 to 2024. We eliminated reviews, case reports, and studies that were off topic, and papers that were out of scope were those on ABO antibodies and SNPs. In this review, we identified 33 papers, and these again have variable hypotheses, study designs, and conclusions. And while our literature review remains ongoing, so far we have not significantly changed our prior recommendations for testing. But some things that we have found interesting and potentially worthy of more study and discussion are these common themes that are also cited in the more recent literature. First, non-HLA antibodies seem to be found in high frequency in multiple study cohorts. And it is also encouraging to find that there are efforts to evaluate microvascular inflammation without HLA donor-specific antibody as an ongoing theme since our last review. We also found several papers on the antigen GSTT1, which is a polymorphic gene resulting in a positive or a null antigen expression and its association with anti-GSTT1 antibody and transplant outcomes. And in addition, there were several papers using non-commercial assays and new methods to identify additional relevant antigens in silico or in tissues. And I want to say again that our review of the literature is ongoing, and this slide is not comprehensive, but this body of literature seems to point to the relevance of non-HLA antibodies and an association with poor outcomes. But the literature is lacking overall in studies that would help deepen our understanding of non-HLA antibodies and clinical outcomes. The ideal prospective data would include a cohort of well-characterized patients tested pre-transplant with no HLA donor-specific antibody or non-HLA antibodies at transplant, and then with serial longitudinal testing and protocol biopsies to determine if non-HLA antibodies can be associated with poor graft outcomes in a model that adjusts for confounders using multivariate statistical processes. In addition, we cite the importance of antibody detection reagents that accurately and reproducibly identify non-HLA antibodies. So currently, there are two commercially available non-HLA antibody Luminex-based detection tests. One of them is from warfarin Immucor, and the other is from 1-Lambda. In the warfarin set, that is one bead set with 60 non-HLA antigens. And in the 1-Lambda assay, there are actually 3 different kits that make up this entire autoantibody detection kit with a total, the warfarin assay has 60 antigens, 60 beads. The 1-Lambda assay has 39. And in common between these two are 25 antigens. But there have been some recent questions and concern about the reproducibility between the two assays, as well as the intra and interlaboratory reproducibility. And these concerns are raised by at least the 2 studies that are entitled here. The first I'll go into in further detail in a few slides. And that one tested the reagents from both vendors. And the second is from unpublished data from the Canadian Autoantibody Serum Exchange, which tested intra-laboratory reproducibility using only the lab screen reagent. So this is data from the first study that I showed on the prior slide. Oh, excuse me, here, pardon me. In a multi-center study, we aim to compare the analytical validity of non-HLA Luminex detection kits from the 1-lambda and the warfarin reagents. This study is modeled on the CEoT 13 study published by Reed et al. That served to achieve similar goals for HLA antibody Luminex detection reagents. We will assess the inter and intra-lab variability of both assays, and we will directly compare the results of antigens shared on both panels. So as an example of the variability and results from the two kits, I have data that is shown here from a set of kidney transplant patients. If we just focus on the panel below, on the x-axis is each bead, and on the y-axis is the percentage of patients positive for antibody to that antigen. Antibodies were determined using both the warfarin and the Immucor assays shown here as assay 1 and assay 2. Assay 1 is in blue, assay 2 is in red, and there is a significant amount of variation in the positive reactivity between the two bead sets. For example, assay 1 shows about 80% of patients positive for GAPDH. The GAPDH bead is right about in the middle of the x-axis. So 8% of patients were positive for GAPDH using assay 1, while assay 2 showed about 20% of patients positive. This data comes from a larger study where the goal of the study is to determine the non-HLA antibody association with kidney rejection in pre-transplant and biopsy serum. Samples from this study are remaining at serum volumes that will permit a multi-center send-out. And we have secured 13 samples that were tested in this laboratory and have been shown to test positive for many of the non-HLA targets. And I'll show you a little bit of these data now. So the goal in choosing the serum for this send-out was to be sure that we had good coverage over the majority of beads in common between the two vendors' bead sets. Here are the data of all of the samples tested on the 1-lambda bead set. And on the x-axis is each of the beads, on the y-axis is MFI. And there's a wide distribution of reactivity amongst all of the beads. So we have negative as well as positive results ranging from 71 MFI all the way up to over 11,000 MFI. And this is the same serum that were tested, the 13 samples tested on the warfarin target. There are 60 beads in this set, and the thresholds here range from 168 to over 8,000. So taking the wide view, this is just the percentage of non-HLA antibodies that are positive in each sample, tested either on the 1-lambda bead set or in the warfarin beads. We have 90% of targets positive in the 1-lambda bead set, and 100% of the targets were positive using the warfarin bead set. And there may be discrepancy due to the fact that the kits and the beads are procured from obviously different vendors that use different manufacturing practices. And there may be different thresholds for calling positive results using both manufacturer specifications. So the aims for our study are to use the kits from both vendors to determine the inter-laboratory variability. So 13 serums will be tested undiluted, and 9 of those will be tested at a dilution of 1 to 4 by 6 laboratories. And then aim 2 is to determine the intra-laboratory variability. 3 of the serum will be tested 3 times by 2 laboratories. In this study, we will test the coefficient of variation at the level of the kit and the manufacturer, between participating laboratories, and at the level of each bead. These are the participating laboratories, and I cannot thank them enough for their participation. Their participation ultimately will help us to feed back to the vendors information on how to improve the reagents. We are currently in the process of obtaining material transfer agreements between Bloodworks Northwest and each laboratory to be able to share the serum, as well as MTAs between One Lambda and Immucor to allow for the distribution of the antibody detection kits to each participating lab. And I wanted to mention that out of scope for this study is lot-to-lot comparison or serum treatments or analysis of the clinical validity or utility of the test. And so just as a bit of wrap up, we know that non-HLA antibodies and antigens have been defined in the literature through multiple studies over the last couple of decades and from multiple investigators. And these have been identified using multiple types of assays and diagnostics, some that are commercial and some that are lab developed. And so through this study, we are working to further develop a deeper understanding of the analytic validity of the commercially available Luminex-based non-HLA antibody detection kits, so that we may further translate current and future hypotheses and conclusions about non-HLA antibodies and transplant outcomes to studies of clinical validity and utility. And I would just like to thank the participants of the non-HLA subcommittee and the participating laboratories, One Lambda and Warfarin for their generous support, and ASHI and AST for their support of STAR. Thank you so much, and I'll turn it back over to our moderators.
Thank you so much, Michelle. I invite all of the panelists for this particular session to put on their cameras. A great session, and I'll highlight that we're really working on analytic validity before we can, think about how we can take this to the clinic. I saw a really good question in the chat. It was that given some of the preparation of these assays is proprietary, will you be able to obtain information on target or source? For example, Vimentin appears to be consistently positive on one of the panels, but negative on the second. And is this due to differences in how the protein is attached to the bead or the recombinant protein?
Maybe just a quick reminder before we're going to have answers from the group. I see a lot of people putting their questions in the chat. Please put them in the Q&A, because otherwise it's really difficult for us to make sure that we're not going to miss your questions. Thank you.
Thanks.
Sorry about that.
Thank you for that question, Carrie, and to whoever posted that question in the chat or in the Q&A. I wanted to say that our collaboration with both Warfarin and Immucor is one of the most important elements of this study. And through that collaboration, we're able to discuss with them and they will give us whatever information that they are able to give so that we can try to understand just the nature of, how the beads are comprised, how they're made. We do know that they are likely sourcing antigens from different sources, and whether or not that is affecting variability within the test, I think we will gain an understanding of that throughout this study. So I hope that if there are similar type questions that, we will be able to collate some of those after this presentation and hopefully address those throughout the study period.
Yeah, one of the other comments was that if the antigen coded on the bead is not the same amino acids, it's really hard to compare. Also, what is the reference or the gold standard in order to compare them?
That's a wonderful question. And I also just want to make sure to allow my co-subcommittee members to chime in. I see Anat and Chris have both opened their mic, and if you would like to.
I can tackle that one, Michelle. I mean, I think to answer your last question in combination with this one, we're not expecting the companies to share any proprietary information with us. And I think, however, it is certainly reasonable that the companies will be interested in what we're doing and use it as a feedback mechanism to improve the assays. And, in that regard, I don't think we're going to find out from the companies whether or not the exact same protein or amino acid structure is present on each bead. But we're going to test the same serum with both vendors, and we're going to find out whether they produce similar results or different results. And that's certainly a step further than we are today.
I would just add that in order to have a gold standard, we have to have, clinical validity. We have to understand if these antibodies are meaningful. But the first step, as Michelle said, is analytical validity. We have to have an assay that is reproducible, and then we move toward finding out what is clinically meaningful.
I think to tag along with this, I'm combining a couple of questions from the Q&A together. But really, people are interested in how a positive versus a negative are being determined, and what are the samples that are being used to make those decisions or to do your wet lab experiments. If you can please elaborate on that. Thanks.
Hi, I can take this one since the samples came from my laboratory. Those are samples for kidney patients, and some of the samples are pre-transplant, some are post-transplant. We tested in our laboratory, and we saw reactivity with both kids, Walandas, and Immunocore, both orphan. And now what we want to reproduce, if other labs are reproducing the same results that we had, so we can compare it. Something like is variability lab to lab, or is intrinsic to the assays, basically. So those are patient samples, kidney patient samples.
And our plan is first to use the manufacturer's recommendations for setting the thresholds and kind of determine the reactivity of the beads and the, how well they have the similar or different results using just those manufacturer recommendations. And then using various statistical models, if we're able, we may try to determine if other thresholds affect those results and what might be best. However, what might be best, again, gets back maybe to having some level of a gold standard and another type of test to cross-validate with, which isn't available that I know of at this time. So I think that our first pass is going to be using the manufacturer's recommendations.
And this, it kind of goes along with another comment slash question that was brought up. The proposed experiments will appropriately address the precision if we're just looking at this assay. But are there going to be future experiments or other experiments to test even the accuracy of the assay? Are there animal studies to prove the pathogenesis of a different, of these different detected antibodies with the assay?
These are great questions. And I think that they also get back to the recommendations that we made in the STAR 22, which is that basic science in this area is needed, that the identification and derivation of animal models or cell models to help us understand the effects of these antibodies, either in culture and in tissues, would be very helpful. There was the other question, future studies we're testing the precision of the assay, but would we test the accuracy, I think was the question going forward, using various samples. And I wanted to mention that one problem that we have encountered through this study is these samples are very precious. There's almost a lack of these kind of samples in the world, and it's difficult to gather samples to do additional studies. And through talks and presentations such as this is how we've been able to be in contact with some other laboratories that are doing work in this area. And so if you have samples that might be useful in the future for these kind of studies, we would greatly appreciate your input.
So there's some really great questions. And I think what we're coming up with is that there's a lot of questions in this area, a lot of work to be done. And so I think as a clinician, we still have to be very, very careful when we get a positive result, because obviously there's a lot of the questions on the lab. So we could spend a lot of time on this, but I think it's time to move on to the next presenter. Again, thanks for these questions. We're going to keep all of this into consideration and incorporate this in the face-to-face in February and also in the manuscript. So thank you. Thanks again to Michelle and the work group for that great work. I'm going to next introduce Marlena Habal. She is a cardiologist at NYU Langone Health, and her presentation is Harmonizing Efficacy of Antibody Reduction Therapy Using Titers. So look forward to this.
Not letting me do anything. Here we go. Okay. Yes, thank you so much for the opportunity to talk today. So I will be talking about harmonizing the efficacy of antibody reduction therapies using titers. I don't have any disclosures, but I would like to take this moment to introduce the study team. So the core members, including Maria and Anat, where we really developed what we were going to do for this particular study group, and then the entire collaborative team, including the members listed on the right hand of the screen. And so with that, I will talk a little bit more about everybody's collaborative efforts in a bit. But what I wanted to do was start by defining what the problem is that we felt we had to address. And I think there's growing recognition that there's really limited standardization in the approach to reporting antibody characteristics. We know that heterogeneity limits interpretive value when comparing studies between centers. And hence there's a need for a way to reproducibly categorize antibodies into different groups: weak, moderate, strong, and very strong. And this led to the STAR 2022 recommendations for reporting standards. And so briefly, I'm going to really focus on those that were derived for desensitization trials. The first being that a CPRA should be determined in at least 2 titers. And I'm going to review what that means in just a moment. But before that, I'll just highlight that in addition to that, they recommended using the number of unacceptable antigens before and after treatment. Importantly, transplantation rates were recommended to only be used for patients with an approved living donor and not for those without a living donor due to organ access disparities. And specifically for those with a living donor, secondary outcomes could include the change in crossmatch, change in DSA titer, and transplantation rate. Importantly, the group also acknowledged that these recommendations needed to be validated. And so before going any further, I'm just going to make sure that we're all on the same page as to this concept of antibody titer. So a couple things. Single antigen bead assays operate in a limited range of what we're going to call antibody strength, which loosely can include molarity and or avidity. Bead saturation, therefore, limits the interpretive value when you go above this threshold, meaning that 2 antibodies that have an MFI of, say, 28,000 can have very, very different strengths. And so one way to overcome that is by diluting the serum and retesting it. And these dilutions can then be used to define an antibody's titer. And so I'll just make the distinction between dilution and titer, at least for the purposes of this presentation. Titer is essentially defined as the dilution at which the antibody crosses a predetermined inflection point, or MFI, which for the purposes of the wet lab that I'm going to introduce a little bit later, we defined as an MFI of 2000. And then titers, as you'll see, can be used to calculate CPRAs at different dilutions. So just a little bit more explanation here. If you perform a dilution series, and then you define a threshold of positivity. As you can see here in this example, these dilutions were done neat, 1 in 4, 1 in 16, 64, and 256. And so this actually allows you to sort of interpolate a titer in between. If you prefer mathematical language, it's essentially 2 to the x, where x equals 0 to 8. And so the STAR recommendation was to perform at least 2 titers so that you could get a sense of this inflection point. And so what I'm going to do is just show you a little example of what I'm talking about here. So as you can see for this A11 specificity, there's this marked bead saturation that goes out to a titer of 1 in 64 before reaching a linear decline out to 1 in 1024. On the other hand, shown in the bottom right-hand side of the screen, A69 has a far more linear trajectory. And so this is really just saying two antibodies with very different strengths can have a similar MFI in meat serum, as shown right here. And that diluting the serum will unmask this. And so essentially, this sort of highlights the need to define a response to antibody reduction strategies, which has loosely been called desensitization in multiple titers. Okay, so just a little bit more explanation and then we'll move on. So in terms of defining a titer, just to provide you with an example here, for this B52 specificity, as you'll see, the first time that the MFI drops below that inflection point of 2000 is at a titer of 1 in 1024. And so then using this interpolated titer in between 1 in 256 and 1 in 1024 is 512. And so the titer here has been defined as 1 in 512. Down here for B13, the first drop below an MFI of 2000 is at 1 in 64. And so the titer was designated as 1 in 32. And we'll talk about some new ways of interpolating titer that we're coming up with in just a bit. But the bottom line is that by doing this for every specificity, you can calculate a CPRA defined by the titer. And this was work done by Dr. Tambur's group. And basically what you can see is the patient here at the top, designated P20, by the time you got to 1 in 4 and certainly 1 in 16, you were seeing the CPRA drop. In contrast, patient P2 down here at the very bottom of this figure had a CPRA of 100% out into a titer of 1 in 4,000. So very different phenotypes. Okay, so with that bit of background, here's what we've been doing for the past little while. We started with a literature review to really ask how has the field changed since the previous STAR iteration. Second, we looked at ongoing clinical trials to predict how things are going to look in the next few years. And then finally, we designed a wet lab, and I'll show you the preliminary data from that. Okay, so literature review. So this was really work put forth by my collaborator, Maria Meneghini, and she reviewed over 34 studies, of which 27 were accepted. What you can, I think, see very nicely here is that the vast majority of these studies were done in kidney transplant. And what we really focused our attention on were the prospective interventional ones, of which 4 were in kidney and 2 were in heart. And so then of those, what you can see is that from an inclusion criteria perspective, most included the use of CPRA. And then in terms of endpoints, they all included clinical endpoints, transplantation or antibody-mediated rejection. And then I think what's particularly notable here is that in terms of the antibody endpoints, you can see that there was quite a bit of heterogeneity, including the sort of elimination of specificities, changes in MFI, change in CPRA, and cross-match. But I think what's really missing is that there was very little looking at CPRA and titer. Now, one thing just to highlight here is that all of these studies would have been designed well before the last iteration of STAR. Then in terms of methodological considerations, what you can see here is that of those studies, most of them provided clear time points at which they were looking at the antibodies. They addressed concepts of proson, and at least some included serum dilutions and changes in CPRA. And so really in conclusion, the description of antibody testing methodology was available for most of these studies, and most of the trials results published since the last STAR consensus included at least more than one definition of antibody response. We also looked at ABMR and post-transplant DSA reviewing 26 studies, of which 21 were accepted. Again, really dominating the literature was those studies being done in kidney transplantation, with 6 prospective interventional ones. In terms of endpoints, they all appropriately included clinical endpoints, but again, we see some heterogeneity in the antibody endpoints, with essentially none looking at single antigen bead titers or cross matches. And so in terms of methodology, again, good use of specific time points, which had been in previous STAR reviews a concern. Many have addressed pros on, and overall, in conclusion, there really has been improved rigor in the description of antibody testing methodology. But I think it's notable that still you see that DSA, MFI, and conversion to values below a sort of predefined threshold remain the most frequently reported antibody-related endpoints. Okay. So with that, we also reviewed ongoing trials. And of the 26, 24 were in kidney. Of the 26, we ended up excluding roughly 12 and ended up with 14 that we looked at more in depth. And in terms of inclusion criteria, the most common was CPRA. None, at least from clinicaltrials.gov, used CPRA in titer or in dilution. But again, these are from clinicaltrials.gov. And then in terms of endpoints, we again had appropriate clinical endpoints, but quite a bit of heterogeneity in how people were intending in their primary and key secondary endpoints to look at changes in antibody. So in summary, from our literature review, we concluded that there was increased awareness of the need to consider antibody strength and overall some improvements in methodological reporting, but that we really need additional work to enhance study-to-study interpretive value. From the clinical trials, I think we can see that the endpoints did reflect better awareness of considerations in terms of antibody strength, but that they're still very heterogeneous and that there's been limited use of CPRA and titer. And then a final point is that there's really limited trials outside of kidney transplantation. And so although they will admittedly need organ-specific considerations, I think it's important to remember that core antibody strength assessment strategies will remain broadly applicable. Okay, and so now in the second half of this presentation, I wanted to introduce you to the wet lab work that we've been doing. So why we designed this study. The use of titers to harmonize interpretation of antibody strength across centers has really not been validated. And so the question then becomes, can we agree on interpatient heterogeneity in sensitization phenotype? And I'll talk a little bit more about how we're defining that in a moment. The second question really arises from work done by others, Dr. Reed's group, that showed that standardized operating procedures can dramatically reduce variability between centers when defining antibody strength based on MFI. And so our question is, can we do the same or even further reduce intercenter variability by using titers? And then finally, whether titers improve standardized assessment of treatment response specifically across centers has not been established. And so really what this question is asking is, can we agree on whether a patient has or has not responded to treatment? And if so, can titers reduce intercenter variability in quantifying that change in antibody strength? Okay, so as an overview to our approach, we designed a multi-center study to evaluate the use of titer as a harmonized approach to defining antibody strength and treatment response. And this harmonization was really a collaborative effort between 5 sites across 2 continents. The PIs are listed in the bottom right-hand corner and have all joined us on this call today. We then identified 10 heterogeneous cases and evaluated them before and after an intervention to reduce circulating antibodies. We did not consider what kind of intervention this was, as this was purely a technical study. And we'd like to acknowledge that the reagents were provided by the vendors, Thermo Fisher and Werfen. Okay, so in terms of our protocols, they were collaboratively established for each of the vendors by the group. We aimed to use the same kits, which we did here in the US, and as closely matched as we could for our European colleagues, the same EDTA and the same positive controls. We designed our standardized operating procedures and our standardized plate layouts. And just to give you an overview of what this looks like for sites, we had 10 cases times 2 time points, so 20 samples. Of those 20 samples, we ran them neat and in 4 dilutions, giving us a total of 100 samples. And then we ran those across 2 vendors. So just to provide some preliminary analysis, the rest of which will come forth at CEoT later this year or early next year. The first question is, can we agree on inter-patient heterogeneity and sensitization phenotype? So to explain what I mean by that, if you look at this case, this was from a single center where we originally defined our phenotypes. You can see this progressive decrease in MFIs with each dilution. And I think this contrasts very nicely with a different case where you see this plateau and then linear decrease. And so what we wanted to do was ask, can we recapitulate at least the general phenotype, irrespective of the absolute MFIs, between different sites? And so what I'll show you is a proof of principle using a representative bead from 3 of the centers. And what you can see here is that there was a nice agreement, again, from a phenotypic perspective, that there's a progressive decrease in MFIs across titer. And the same here, although there was some heterogeneity, I think we can all agree that the 2 graphs here in the upper left and bottom right look different, and that all the sites would have had a similar agreement that they looked different. So this was a very qualitative first pass approach. Then we wanted to do something more quantitative, and so we're asking, can we reduce intercenter variability using titer? So this is the overall workflow. We took a sample, we got a neat MFI and defined the coefficient of variability between that MFI between sites. Then we did our dilutions, and we defined titer by interpolation as a continuous variable. So essentially we took the MFI of the titer above and the titer below, drew a line and calculated the slope. And then we took the coefficient of variability between sites doing it this way. I'll explain a little bit more what I mean by that. So if you recall earlier on, I was describing a whole integer interpolation approach as outlined in this figure with the interpolated titers in blue here. What this gives you is essentially a 2 to the 5 or 1 in 32 titer for specificity X, or perhaps I should say A here. And then for specificity B, it's 2 to the 6 or 1 in 128. So that's the approach that we had previously used. But what we wanted to do was see if we could design a different approach. And this one is interpolation as a continuous variable. And so what you can see here is that using this slope-based approach, we actually get a titer of 2 to the 5.7 or 1 in 52 and 1 in 84. And so to compare these two, using the whole integer, your titer would have been 1 in 32, and using the continuous, it was 1 in 52. And here it's 1 in 128 versus 1 in 84. So essentially your titer can either be higher or lower than the whole integer. So using this, we then have very preliminary data from 3 sites using a total of 10 samples. And the bottom line is this is still very much a work in progress, but we were able to show that there were similar coefficients of variability for MFI and titer using these standardized operating procedures across these 3 sites. if you notice in red here, there are a couple of coefficients of variabilities that are really quite high. We've subsequently identified that these are all coming from very, very low MFI beads. And so we're doing some additional analysis to address this issue. And that's essentially further analysis is ongoing. Okay, and so to get to our third and final question of defining treatment response, or can we agree on whether a patient has or has not responded to treatment? Those harmonization exercises are still ongoing, but just to give you an example of how we will be doing this from a CPRA and titer perspective, you can see that this is one of the cases before being treated, and this is that same case after being treated. And so we will ask how much the coefficient of variability changes in that interpretation between centers. So in summary, our working group across 2 continents has been set up to establish proof of principle. Can titers be used to harmonize interpretation of antibody strength across centers? We aim to define new computational approaches to interpolating titer that will require further validation. And in ongoing work, we're testing whether titers can enhance our interpretive value of antibody phenotypes and treatment response across these centers. And we look forward to providing our further analysis at CEoT. And so finally, just to acknowledge, first and foremost, all our collaborators, but in particular, Dr. Jerson, who's been doing the statistical design and analysis for us, Andrea Kim, who has been critical in organizational management, and also all the technicians that performed these assays in their respective laboratories. Of course, from an industry perspective, One Lambda and Warfarin and the AST. Thanks.
Thank you, Dr. Habal. That was a great talk, and it's really nice to see in both talks how the wet lab component has been, added to the STAR work. Again, invite all of the members from this working group to get on to answer questions. One of the questions I think is just really to clarify is, can you explain again how CPRA will be calculated based on the titer? And I'll take this from anyone, and I think it's just a key concept for our listeners.
I'll be happy to do that, Marlena, if you wanted maybe to.
That's fine. Do you want a slide to show it?
Yes, I wanted to ask that the one slide from the paper that we have with Carrie and Mark Stigall, because I think that would be the simplest one to answer for that. So we've been using this for quite some time, and I really want to highlight actually that Mark Stigall was the one who came up with the idea of trying that approach, and the idea was to try and take very complex data and kind of collapse it into something that is measurable and clinically applicable. So I'm referring to the right side of the slide that came up from that particular paper that I'm mentioning right now. As you can see on the x-axis, we actually went ahead and did dilutions much farther than what we've done for the STAR project, because some of our samples were extremely the antibodies for some of those patients were extremely high level, or Marlena used the comparison with molarity, which really resonate with me very much. The principle is really simple. You will determine what is the threshold that you feel is relevant for your center. For our study, it's going to be 2,000. Actually, for this particular study, it was 2,000. It can be lower than this if your center is less averse for risk. It can be much higher than this if you are talking about thoracic organs and you're willing to cross higher antibodies. But the bottom line is you need to calculate the CPRA multiple times. You take the positive results from each of your dilutions. So you can take your neat results and calculate CPRA based on this. You can take your positive results from the 1 to 4 titer and calculate the CPRA based on this, and so on and so forth. And by doing this, what you can see is that at some point, usually there is a big drop in your CPRA. You can determine where you want to see that drop. We were doing that particular study for kidney patients. So from our perspective was to try and see when is the difference from 99.5 rounded up to 100% to 99.4 or below, which based on previous data means that that recipient will have roughly 10 times more compatible donors. And you can, again, do the calculation based on the organ that you wish. So multiple calculations of CPRA for each of the dilution points is really what we refer to when we're talking about CPRA per titer. I hope that answered that question.
And if I could just add something here, more coming from the clinician, that this was a really great way for our patients who all had 100% CPRA to really see how different they are in terms of this antibody strength concept. So not only in the lab, but really clinically, it really illuminated, as you can see in that figure, how heterogeneous patients really are, even though that one number can look the same.
Yeah, and I'll add to this just one more thing. Obviously, is the one who keeps on pushing to doing titers. I do believe, and I hope that came clear from prior presentations of that topic. We'll see what will come up from that exercise. I'm really happy that we're doing it. That doing those dilutions is really going to be beneficial for the more sensitized patients that have low-level antibodies, or a small breadth of antibodies may not, unless we're talking about this high molarity, will probably not going to benefit from this added work, and it won't be relevant for them. But when we're getting into really highly sensitized patients, that's where we felt it is beneficial.
Excellent. There was a question, are you using several antibody panels or only one from the most common vendor for the wet lab?
Marlena, do you want to answer that? We're using both vendors. I don't know what is considered the most common. There is one vendor that is more used in the US. There's the other vendor that is more used in Europe, I believe.
Sorry.
Go ahead, Marlena.
No. But was the question, are we trying it with different lots or different vendors?
The question was about vendors.
Oh,
and
okay.
they had
Sorry.
the specific question is, are you using several antibody panels or only one from the most common vendor?
But antibody panels meaning like different beads, or?
I think it's hard to know based on this, but I think just explaining exactly, what you're doing, that you are doing it on this using two different vendors.
Yeah, and one of the ideas to get laboratories from Europe was really to try and make sure that between Europe and at least North America, regardless of the vendor that you're using, we can all talk in similar language, with the idea that obviously in both continents, people are reporting on studies, and people would like to learn from those studies and see if they are applicable for their own center.
Okay, I think we have one more question and then we'll move on. This one was, thanks, Marlena. When you calculated slopes, were they based on specific beads as similar as shown in your example slide, or based on all combinations of CPRA of 2000?
Yeah, that's a great question. No, it was done individually for each bead.
Great. Very interesting talk. Great progress from your working group. So let's move on. I will now invite Anna Morris, PhD from Emory University, and she's going to talk to us today. The title of her presentation is Epitopes, Eplets, and Transplantation, Quo Vadis.
Yeah,
Thank you, Carrie. Yes, thank you for having me today on behalf of our working group, which includes Dr. Howie Gable, Sebastian Height, and Anat Tambur. And so today we're going to be talking about epitopes, eplets, and transplantation. And the Latin Quo Vadis means where are you going? And I'm using that colloquially as where are we going as a field in terms of epitopes, eplets, and transplantation. And so as a brief way of introduction, as clinicians, you might have been inundated with matching algorithms in the last 5 to 10 years, which each seek to understand whether we can identify low-risk transplant pairs and possibly minimize immunosuppression. So one major avenue of this has been through eplet matching or molecular mismatch load analysis. And so STAR actually mentioned the concept of molecular mismatch load back in the 2019 report. With the gap that we needed tools to prospectively determine immunogenicity beyond the molecular mismatch load. And then it was mentioned in the STAR 2022 report, the recommendation was to assess mismatch load as a biomarker. Then the gap remained that we needed to better understand immunogenicity and assigning of permissible versus non-permissible mismatches. And so now with these gaps highlighted in the rear view mirror, the field continues to charge forward. And we would just like to take a brief pause in this moment of innovation to think about the definitions of these terms based on a historical understanding of antigen and antibody recognition and responses, and then make a few recommendations for our clinical and research sides. And so to begin, I've made a few quick poll questions, and unfortunately we can't actually poll you today, but I just want you to think of the answer in your head for now. So what is an epitope? Is it a small region of non-self amino acids? Is it 2 to 5 non-self amino acids within 3 to 3.5 angstroms? Is it bound by the paratope? Is it immunogenic? Is it helpful for matching? Is it more than one of the above, or is it none of the above? So I'll give you just a few seconds here to think about your answer here. What is an epitope? And I'll tell you that the closest answer is C. It's bound by the paratope. It's the entire contact region of an antibody or the paratope to an HLA antigen when we're specifically talking about HLA. Okay, and so my next poll question is, what is an eplet? And so there we have similar answers for this question. Small region of non-self amino acids, 2 to 5 non-self amino acids within 3 to 3.5 angstroms. Is it bound by the CDR3? Is it the immunogenic part of the epitope? Is it helpful for matching? Is it more than one of the above? Is it none of the above? Again, I'll give you a few seconds to think about this one. And so if you're thinking that it's maybe more than one of the above, you're probably aligned with the rest of the field. However, we'd like to put forward that this is a term that's restricted to HLA that's been put forward by René Duquesnoy to describe the potential target of an antibody within an epitope. And so it has been described by B here, 2 to 5 non-self amino acids within 3 to 3.5 angstroms, but you've probably seen it as talked about as it's bound by the CDR3, it's the immunogenic part, and it's helpful for matching. So you might have seen all of those being talked about in terms of eplet. And so as we thought about these complex topics, it became apparent that something we use every day has actually many parallels to antigen recognition, and that's facial recognition software. So over 131 million Americans use this daily. And so what it does is it takes measurements between facial landmarks, between all of these facial landmarks here, and then creates a code for each individual. And so when you go to open your phone, it essentially makes those same measurements and ensures that your code is coming up when you try to sign into your iPhone. And so as we have been through the pandemic, as we just went through summer, we know there's actually many woes with facial recognition. So if you are wearing a mask, you might not be recognized by your phone. If you're wearing sunglasses, you also might not be able to gain access to your phone. And that's because many landmarks are critical for recognition. And if not all are met, there's no recognition. And that's critical for its specificity, meaning that siblings with your eyes cannot access your iPhone, and maybe a parent or child with your smile cannot access your iPhone either. And so when we think about antigen recognition, what are some parallels that we see? Well, in the yellow and blue is our antibody bound to the green antigen. When you peel them apart, you begin to see that there are these red amino acids that are the contact regions of the antigen and the antibody. And so many areas, again, are critical for recognition, and if not all are met, recognition is compromised. And that includes these amino acids contributing to complementarity and chemical contribution. And then if you copy and paste only a certain region of these amino acids onto another antigen, it's likely going to be insufficient for recognition. And so we do see some parallels there with our facial recognition software. And so the status quo of HLA antigen recognition has been built for the last 20 years by René Duquesnoy, and it was a novel paradigm-shifting idea that polymorphisms could be assessed in triplets, and sharing of those triplets between a donor and recipient despite HLA antigen mismatching could identify compatible pairs. So this could be used for highly sensitized patients to identify whether a given donor would be compatible based on matching at triplets in which they would make no antibodies. And so this was the HLA matchmaker tool. However, as human nature, we wanted this tool to do more, and so it morphed into a prediction tool for antibody production. Instead of assessing what triplets were shared, now those triplets evolved into Eplets that were then assessed for the ability to induce an immune response. And with this came a new vocabulary for B-cell epitopes based on hypotheses that turned into assumptions that were then accepted by the field. And so as you can see here, we have Eplet being defined as the center of an epitope with a dominant role in determining antibody specificity. It was also called a functional epitope. It was 2 to 5 amino acids within 3 to 3.5 angstroms. It was suggested to be bound by the CDR3 of the antibody. It was suggested to require amino acid polymorphisms to make it immunogenic. And then that region surrounding the eplet or functional epitope provided a radius, provided the structure. So that was within 15 angstrom radius. And so this vocabulary differed from original B-cell epitope literature, and for somewhat good reason. Viral and bacterial surface proteins are completely different than self, and so they form epitopes that differ greatly than self. However, transplantation of foreign HLA results in much more minor differences than self. And so you have A1 here, and the amino acid polymorphisms that differ to A2 are shown in red. And so instead of the entire viral protein or a bacterial protein being made up of foreign material, now we only have certain amino acids that now look different than self. And so that allows us to now ask very particular questions, like which amino acids are critical for antibody binding, which are critical for inducing the immune response, and can we identify acceptable and unacceptable mismatches? However, assumptions were made that epitopes can be subdivided into specific regions with size limitations, with functions. And these assumptions were not based on previously known characteristics of antigen-antibody interactions. And so we would like to posit that assumptions with lack of evidence may lead to pitfalls in analysis. And 2 pitfalls I'll give you today is 1, over and under interpreting antibody responses based on Eplets or certain amino acid polymorphisms. And then 2, assuming that immunogenic in one individual means immunogenic in all individuals. And so I'll first tackle this first assumption. And really, I'm pulling my example here from this paper down here from 2022. And so if you look at this single antigen bead assay, all 31 FYY eplet-bearing alleles are to the left of this red line. However, to Did I say left? Yes, I did. That were two. However, they all differ in range of MFIs, with half being very high and half you can see decreasing drastically in intensity. And so over-interpreting this antibody response could lead to the assumption that 31FYY is the major target of the antibody, and all specificities that bear it, even if it's below our threshold for calling as unacceptable, should be called as unacceptable, and we should block any donors for this recipient that have those antigens. And so I would give you this analogy. Could be akin to tossing the baby out with the bathwater. Another interpretation would be under-interpreting the antibody response. So it could be assuming that 31FYY, because there's so many various MFI levels associated with it, this possibly cannot be the target of the immune response. And so maybe you only call the highest MFI antigens, or worst case scenario, you interpret this as non-specific binding and then call nothing as unacceptable, which could be very detrimental to your patient. However, when we assess the amino acid polymorphism surrounding that 31FYY eplet, shown in yellow here, it becomes clear that polymorphisms outside of it are capable of altering the binding of the antibody and lowering the MFI intensity of these particular HLA antigen-bearing beads. And so over-interpreting could lead to blocking of antigens that are unnecessary. And again, under-interpreting could lead to crossing very real HLA antibody. Okay, and so to tackle the next pitfall that we might fall into, that immunogenic in one individual means immunogenic in all, you might assume that a patient cohort with the same eplet mismatch has the same risk of developing an antibody response. However, this does not take into account the physiochemical properties of that mismatch that may differ in the setting of different HLA alleles, and differences to the self-HLA of that individual can influence whether that eplet is seen as immunogenic as well. And to give you an example, this is from a 2014 paper looking at electrostatic potential. All of these beads have the same eplet. And if you look at the ability for that electrostatic score to group together, you can see that all of the antigens that bear this eplet actually fall into two different groups, the red box group and this green box group. Therefore, a mismatch in one individual with a certain HLA molecule may not carry the same risk, maybe much lower risk in this green box, compared to someone else with the eplet, another HLA molecule, say one that is in this red box here. And so how do we combat these pitfalls? Well, a few things that we think we can do is, one, going back to the basics, and two, collecting appropriate data. And so to go back to the basics a little bit, what do we know about the B cell epitope? In the 1990s, there were very few crystallized structures that were known, but still remains that crystal structure is the most preferred way to study B cell epitopes. And so at the time in the 90s, you can see that we have our paratope in red here, and then our epitope also in red. And the assumption, or hypothesis, was that about 15 to 22 amino acids made up those surfaces, and that the complementary surface was about 600 to 900 angstroms. Well, now in current papers, there's over 800 plus crystal structures of paratopes and epitopes able to be assessed. And these data mainly confirm those findings from those original crystal structures that were identified, that the average touch points are about 18 to 19 amino acids on both the epitope and the paratope. And those amino acids contribute complementarity, hydrogen bonds, salt links, and hydrophobic interactions. And on the paratope in particular, on that antibody, or the BCR, 90% of interacting residues are within CDRs 1 to 3, and approximately 60% are non-CDR3 contacts. And so making the point that there are residues that are outside of CDR3 that are making contact within that paratope. And so a concept that's been talked about with epitopes since the 1960s is immunogenicity, or the ability to generate an immunological response. And so what do we know today about immunogenicity? So first, factors that are innate to the epitope and to the paratope are both critical for appropriate antigen recognition. And so on the epitope, there are residues that contribute to contact, so those that contribute to shape complementarity. There are those that contribute to energetics of binding, so those that contribute to chemical complementarity, and then those that differ from self, and so amino acid polymorphisms. Those all contribute to the epitope. And then for the paratope, we also must make, a complementary shape and chemical complementarity to match our epitope. But on the paratope, these are influenced by the HLA genotype of the individual, which will then influence naive T and B cell development, and previous immunological experience as well, which will influence the T and B cell repertoire of an individual as well. And so these factors describe antigenicity, or the physical properties that allow for paratope to epitope binding. Now, if we think about immunogenicity, or the ability to induce an immune response, not these factors of the epitope and the paratope here are subject to biological properties of the system, such as the presence of T cell help, and the strength of exposure and the sense of danger associated with that exposure. And specifically when we think about transplantation, we also have immunosuppression on board, right, which influences the biological system that the epitope and paratope are seen in. And so with consideration of these biological properties, we now can begin to understand or study whether a particular paratope or epitope interaction will lead to an active antibody response and can be termed immunogenic. And so immunogenicity, again, is that ability to generate an immunological response, and it involves these biological properties acting on the epitope and the paratope binding. And so immunogenicity then is not solely based on foreignness. And so our zoomed in approach the last several years, even decades, in studying HLA epitopes via only these foreign polymorphisms or mismatches is too narrow, and based on the fundamental, based on those fundamental concepts of immunogenicity we just discussed. And so instead, if we adopt a zoomed out approach in which we consider an entire epitope and its contribution to paratope recognition, and in addition to those biological properties that then act upon it, that now allows for more accurate view of the gaps we face in understanding HLA recognition. And so when we do that, when we scale out, we consider HLA genotype that becomes paramount. Potentially surrounding amino acids that contribute both shape and chemical complementarity are considered, other than those that just differ from self-HLA. It also expands our understanding of amino acids that could contribute to the epitope, like peptides within the binding groove, within that peptide binding groove, if they're within the contact region, or the alpha chain of certain class 2 molecules as well. And so moving forward, we would like to posit some more accurate use of terminology. That epitope considers the entire contact region of an antibody to an HLA antigen, and that further amino acid polymorphisms that are shared by one or more HLA antigens could provide important binding energetics for an antibody. However, other amino acids in that epitope can contribute and are critical to antibody recognition. And it doesn't mean that those polymorphic residues are in the center of the epitope either. And then when we think about immunogenicity, these are contributed to by epitope and paratope properties, as well as the biological properties of the system that act on those. It cannot be defined by single antigen bead reactivity, and cannot be a single amino acid polymorphism. And it cannot be extrapolated to the population without cohort data and high resolution typing data. And so where do we go from here? We have some clinical recommendations. And so again, STAR 22 suggested that the molecular mismatch load analysis should be assessed as a biomarker because we don't currently understand that complex contribution of the entire epitope on immunogenicity. And so with that in mind, it shouldn't be used in isolation for clinical recommendation. But it's one of many tools to hypothesize production, antibody production reactivity. And then we also want to make recommendations for how to pursue research in this area. And so there are several levels in which all labs can contribute to. And so first, when assessing antibody reactivity, include as much descriptive information as possible. So the types of descriptive information is listed here, which includes crystal structures or 3D modeling if that's not possible, amino acid alignment, those that are involved in binding, electrostatic properties, and especially two-field typing of the donor and recipient. Secondly, provide evidence of an immune response via experimental verification. And so there are several papers that right now that include guidelines. So I've included those guidelines down here. And so this is also going to be a focus of the 19th International Histocompatibility Workshop. And so more to come on how labs can contribute experimentally to verify immune responses against potential polymorphisms and epitopes. And then finally, we can provide functional data via cohorts that have graft outcomes associated with that. And that is really looking for the evidence of clinical impact due to antibody responses against particular amino acid polymorphisms and epitopes. And we also encourage international involvement. So again, I mentioned discussion at the International Histocompatibility Workshop, and for discussion there to establish accurate terminology for these complex biological states and appropriate avenues. And as we've said, HLA has a very particular niche to study this outside of any other field in immunology. And so we really have a great opportunity. And then an avenue to publish new antibody-verified polymorphisms based on clearly defined levels of evidence. And so that could be verification alerts in all of these journals that just make it easier for labs to publish their data about antibody responses. And so with that, that wraps up our discussion so far in this working group. And so we'll take questions, comments, and feedback. And really just want to thank everyone in our working group that has participated. It has been such a fruitful and insightful discussion, and looking forward to your feedback now. Thank you.
Yeah, thanks, Anna. Very detailed, interesting topic. I invite all of the panelists to get on. One question, and this is really for anybody, is, ultimately it depends on how the patient does long term. That's why we do all of this work. And there's multiple, algorithms that are now published, available, marketed. Have we really compared, how well these different algorithms work, or are we even ready to use these algorithms?
That's a great question. So there have been studies that have compared different algorithms. We can look towards Chris Weeb's group, Peter Nickerson's group. They've compared several algorithms to look at low risk mismatch loads, if that's what you're referring to. Are we, like we mentioned in previous stars, these mismatch load calculations are great biomarkers to kind of parse patients into different groups to allow for associated risk to be known. However, we don't currently, we're not able to look at immunogenicity of particular Eplets and what that means. So you might have a low mismatch load. However, you might have a particularly immunogenic Eplet that may lead to a potent immune response. And so I think that that is still to be worked out. Does anyone else want to comment from our group?
Yes, if I may jump in a little bit, I think we need to, ask the question is, what is the use that we are interested to use it for, right? I mean, is it gonna be to determine organ allocation? Is it gonna be to determine whether to put a patient on a particular immunosuppression protocol, and so on and so forth? So I think those are different questions, and what you're looking for is probably your question will, I guess, direct the answer that you're going to. And Anna alluded to the fact that we have, and Peter's group had shown beautifully that using Eplet can be used as a biomarker. The question is a biomarker on its own sufficient to make a sole decision to make, or is sufficient to make a decision? And I've brought multiple times the issue of looking at prostate cancer. If you have a particular PSA result, is that sufficient to go ahead into surgery? On the other hand, I think the data that they had provided showed really nice correlation with antibody-mediated rejection, T-cell mediated rejection, and I believe, and Chris may want to talk to that effect, that they are trying to recommend to use this for determining protocols of immunosuppression rather than organ allocation. So I think we need to be careful. And one of the things that I would say STAR overall is trying to make sure, at least that was the original intention, is to educate all of us to use the right terms and the same terms to address a particular issue. And then if we want to talk about molecular mismatch load, or if we want to talk about immunogenicity, or if we want to talk about biomarkers, we need to understand what we're talking about. If we're talking about Eplets, and if we're talking about epitopes, and I think this is what Anna was so beautifully trying to show us today, Eplets and epitopes are not the same thing, and therefore we shouldn't expect them to answer the same question. So I see more hands up there, so I'll be quiet.
And there's also another question that we have in the Q&A. So Eplets are being identified and verified using artificial systems. Is that a concern when we want to eventually move to clinical utility?
Well, like we mentioned, we cannot read out immunogenicity based on single antigen beads alone. And so I think that is something that we must keep in mind when we move forward, especially too, when we think about our functional cohort data. I think that's why cohort data is so critical, that we have a group of individuals that have potentially that same eplet, that then some make that antibody response or others don't. And so, yes, Anat, do you have more?
Well, I think you've mentioned, you addressed this in your talk, and really the need to do absorption and elution experiments and using actually cell-based assays rather than bead assays is probably going to be really important to highlight the issues associated with the three-dimensionality of the molecule. So I believe Anna had addressed this really nicely, and I see Howie's hand up there.
Yeah, thanks. I just want to come back kind of full circle, and I think the theme of each of these presentations today is the evolution that we've undergone from the beginning till where we are right now, and that each step has been a step forward and a huge progress. And now we have new data to reevaluate things that we interpreted only one way in the past. And I think we should look at this and take into account all of this new information, a new way of looking at things, and help us move forward to the next level, which is going to be basically ending with what Anna did today, what I think is a superlative job of trying to get us all onto the same page of what we consider an epitope and an eplet and what's immunogenic or antigenic. These are the questions that can be addressed now, but we wouldn't have gotten here without all of the other data that's supported us yet as far as we have right now.
Yeah, 100%.
Well, I think here we're getting close to the end of the session. So if you want to provide maybe more information about the next webinar and the in-person.
Sounds good. So I hope, thank you everyone for joining. I thank the speakers, the panelists, all the working group members for really putting a lot of hours in to get to this point. I invite everyone to join us again on Thursday, same time, same place. We have 2 talks, one on cell-free DNA, the other on innate immunity, and again, we'll have a lot of time for discussion. I know that there, may come up with, the audience might have other questions, comments that they have. Please feel free to, reach out to us. And I think that Brian or AST, even if you reach out to them, they'll get us that information. And we're happy to incorporate that in our final manuscript and work, and a commercial for the in-person STAR meeting in February, just prior to the CEoT meeting. So thank you. I'm not sure if you have anything else you wanted to say, Anat.
I just put my email address in the chat for everyone. If anyone felt that they had not had time to put their questions or something pops up later, please feel free to send it to me. I'll make sure that I'll direct it to the relevant groups. We really want to have those webinars in order to solicit feedback from you guys, and then the individual groups will discuss this and decide how to incorporate this. And I believe Brian Valeria, our host, wanted to have the last parting words. So Brian, the stage is yours.
Well, thank you so much. AST and ASHI would like to thank our STAR working group presenters and panelists and all of our attendees for today's webinar. After we close the session, please take a moment to fill out the evaluation survey link to help us keep our content current and engaging, and to help the STAR workgroup members prepare for CEoT 2025. Please remember to join us at 1 p.m. Eastern Time on Thursday for day 2 of the STAR working group webinars, as Dr. Schinstock mentioned. Your brand new login link to join us on day 2 will be emailed to you tomorrow. Everyone registered for today will receive one new link. Thank you all again for today's session.
Thanks.